Hi dear students, a warm good morning to all of you. Today we are going to talk about the topic microbial taxonomy. Before we define what is taxonomy and all that, let me just ask you some questions. Can you identify these organisms? Of course, you will be able to identify them as monkeys, parrot as well as penguin. Without any hesitation, you answer them. Now, why do you answer them? It's because you have already set in your mind that if such characteristics are there, you call the organism as as a monkey. And when you see some phenotypical characters like the green feathers, the red beak, as well as the eye, and all the shape of this, you call this bird as a parrot. And of course, the same is the case with the penguin. So, though you are not easily responsible for you are not the one who have made up the scheme and all that, our ancestors previously had made up a guideline to characterize the organisms, name them, as well as classify them. And this science of what biological classification is called the taxonomy or systematics, and it mainly consists of three separate parts: classification, nomenclature, and identification. In order to classify the microorganisms, okay, both micro as well as macroorganisms are being classified. And in order to classify them, we use a particular scheme. What is a scheme? A scheme is a guidelines saying that all these characters are needed in order to group the organisms into small groups called the taxa, or the taxon is the singular form. And this is mainly based on the mutual similarity that we divide the organisms into taxa. And after dividing them into taxa, you also name them with specific names. That is, you give each taxa a specific name. That is called nomenclature. And later on, when you see any particular isolate which has which is satisfying the classification scheme, you identify that particular isolate to belong to a recognized taxon. This is called identification. So the biological science of taxonomy, it mainly consists of three different things: the classification, nomenclature, as well as identification. The first classification now which is being followed is the natural classification, which was being developed by Carolus Linnaeus, and this was mainly based upon anatomical characteristics. And you can see that it did not give any much information on the evolutionary relatedness of the organisms. So there are lots of classification systems, and the first, or the natural classification, mainly was mainly based upon the anatomical characteristics. That is, how do you know what is anatomy? I'm not going to the details of that. So when a classification system was being developed based upon anatomical characteristics in the 18th century by Carolus Linnaeus, we classified organisms, and that classification is called the natural classification. And in some cases, you can also see phylogenetic classifications. What do you mean by phylogenetic classifications? Phylogenetic classifications are the classification systems where we use the phylogeny or the evolutionary relatedness of the organisms. I will just give you an example. Okay, we have a what? When we see human beings, we call them as, okay, Homo sapiens, we give the scientific name, or the humans we call them. And if we trace from where do we these humans have been originated, we have a ancestor related to a monkey. So such case where you find out the evolutionary relatedness or the ancestral development of a particular species, that is called the phylogenetic classification. And usually, such direct comparison of genetic material and gene products will aid in this. Now, in the case of woos and fox, they use the small subunit ribosomal RNA nucleotide sequences to assess the evolutionary relatedness of the organisms. And some classification systems also use the genotype. That is, the genetic similarity between the organisms can be used to identify whether what is the percentage of similarity between two different organisms. So you take individual genes or the whole genomes, and then you might compare that between each other. And if you say that 70% of homologous regions are being found, homologous means the ones which are being found to be more of similarity. In such cases, we consider them to be of the same species. Another thing which I need to talk about is a polyphasic type of classification. I have not included that in my slides, but let me tell you that. Right now, we use a polyphasic type of classification in which multiple levels of things have been considered for classification. And this is also another classification called the phenetic classification, where the mutual similarity between the phenotypes or the external morphology is being taken for as a guideline for the classification of organisms. So when we consider the classification systems, the first one was a natural classification. We have the phenetic classification, where the phenotype or the mutual similarity between the phenotype is taken as a basis of classification. Then the phylogenetic classification, where the evolutionary development is being noted. The genotypic classification, where the genetic similarity between the organisms have been taken into consideration. And also the polyphasic taxonomy, which includes multiple factors, like it could be phenetic factors, it could be phylogenetic factors, it could be genotypic factors, and all that. So that polyphasic taxonomy is a one which is being adopted right now to classify the organisms. Now let's talk about the levels of classification. In which order do you classify the organisms? As I mentioned earlier, a group or the level of the classification is known as a taxon. And we have arranged different taxon in a hierarchical order. Now, this is the most common type of hierarchy, the kingdom, phylum, class, order, family, genus, and the species. Uh, Kingdom was like we use it for the plants and the animal kingdom, but we do not use it most for the my bacteriologists. And phylum, that of course we use it for botanists as well as for bacteriology, or for the microbiology also we use it. So this order of the levels of classification is important. I guess you are clear with it. Now let's talk about the taxonomic ranks within bacteria. Now, all the principles of taxonomy is also being followed in the case of bacteria also, and they also have taxonomic ranks. But the highest rank of bacteria comes under domain, and after domain we have the phylum, class, order, family, genus, species, and all that. So you can just have a look of it. That will be more helpful for you. You have the domain. Instead of the kingdom, you have the domain, phylum, class, order, family, genus, and species. So domain bacteria, then comes phylum proteobacteria here, then alpha proteobacteria, beta proteobacteria, gamma proteobacteria, delta, and epsilon, and in that you have divided. So unlike the kingdom which was been given here, in the case of bacteriology we have the hierarchical, we call it as a domain instead of kingdom. So these are the taxonomic ranks of bacteria. Now let's define, how do you classify organisms as a particular species? The basic unit of taxonomy, representing a specific genus type of organism, is referred to as a species. And we call those organisms which are being reproductively isolated or which are reproductively compatible, we call them as species. Now, what do you mean by that? We know that I can explain it with an example. We know that dogs do breed only with dogs. Dogs do not breed with cats. The same is the case with different all other organisms of different species. So, if a group of organisms are being found to be reproductively isolated or they are only able to reproduce with their own type, then we call them as, of course, and they should be similar in nature also. That is quite common. If the organisms are being found, considered to be as having similar characteristics, and if they are being considered to be reproductively isolated with their own type, then we call them as species. That is a common definition that we give for the higher organisms. But we know that many microbial species, including bacteria, do not reproduce sexually. So in such a case, the definition or this definition cannot be given for what? The bacteria. So how do we define species? So species in the case of bacteria is a collection of microbial strains that share many properties and differ significantly from other groups of strains. I will go into the details of it. Just remember, it is a collection of microbial strains which have many common properties and they differ from what other group of strains. And the species is being identified by comparison with a type strain, a well characterized pure culture, or the reference for the identification of unknown. Now you have come across a term called strain over here. What do you mean by a strain? That is important because unless and until you understand what is a strain, you cannot call it understand what is a species. A strain is nothing but a bacteria which has been come from a single pure microbial culture. Do keep in your mind, in the natural environment, bacteria do not live individually. They would be living in association with a mixed population of bacteria. Now, if you isolate them into a single type of bacteria, that is referred to as making it pure. And the strain refers to all those bacteria which have been isolated from a single pure microbial culture. That is, on the agar plate, you might get one particular dot of a bacterial culture. Okay, that is, and all the descendants from that particular dot of the bacterial culture is referred to as a strain. And you can look when you isolate soil, bacteria from the soil, you get different particular types of bacterial strains, which are because they would be of different characteristics. So, if a group of strains share a similar properties, then that is a microbial strains. If a collection of microbial strains share what similar properties, and if they differ from other strains, then we call them as a species. Clear? And so the strains itself, now how will they be different from others? They can vary in depending upon their biochemical characteristics, or sometimes morphologically, or sometimes by its antigenic properties. So if the strains vary based upon their biochemical properties. Now, what is biochemical properties? Let me just explain. Sometimes bacteria can utilize sugars. Some bacteria can use lipids. Some bacteria can use pesticides. Like that, some can produce acidic end products. Some can produce alkaline end products. So, because of the metabolic diversity, okay, the biochemistry of the organism or the biochemical properties as well as the physiological properties. Now, physiological properties means some can use oxygen, some cannot use oxygen, some can use carbon dioxide. Like that, based upon the biochemical and the physiological properties, some strains would be different. They are referred to as the biovars, and morphologically they will be different. Now, some bacteria will be appear to be as green in color, some will be yellow, some will be green, red. Like that, on the morphological features they would be different. Such strains which are different for their morphological characteristics are referred to as the morphovars. And then we also have some of the strains which differ in their antigenic properties. What do you mean by antigenic properties? On the surface of COVID virus, you have a spike protein. The spike protein is something which is unique to the COVID virus. Okay, so on the surface, and the speciality of this spike protein is that it is something which is defined or which is present on COVID-19 only. So it serves as an antigen. Okay, and we produce antibodies against these antigens. So, different strains of bacteria. Now, I mentioned a virus example, but the same is the case with the bacteria also. So, if you take a bacteria, on the surface of it, there are certain molecules. They serve as the antigens. And these antigens are the ones which will induce the production of antibodies within our body. So, if bacteria differ in their antigenic properties, then we call them as the serovars. Now, another term which we come across is the type strain. What do you mean by a type strain? The type strain is the first strain of a species which we have studied. Do remember that now I'm going into the details of microbial taxonomy in depth, because the strain and all that comes only in only in microbiology. So the first strain which was being studied or which was being fully characterized is referred to as a type strain. And not always it could be the first strain, because sometimes it would be the more fully characterized strain which will be considered to be as the type strain. And it not necessarily most representative member of the species. It would be usually not representing, but it would be the first one which was characterized or the one which has been easily characterized. Now comes to the definition of genus in bacteriology. It is defined as a well-defined group of one or more strains which clearly separate from other genera. And of, often there do exist some disagreement among taxonomists about the assignment of a specific species to a particular genus, and based upon that, we classify the bacteria into genus also. That is, if some group of strains can be separated from other genera, then we like they have some properties unique to them, then we call them as a genus. So this is the case of classifying the bacteria into different taxonomic ranks: the domain, phylum, class, order, family, genus, and the species. And you know that under the species we do have the strains, but and it is the strains which have similar properties are that which are being considered to be as the what? Species. And after classifying them, we give them names, and that is called the nomenclature. And in the case of bacteria, similar to that of the scientific name, we also give them a generic name as well as a species name. If I consider the example Bacillus and Subtilis, Bacillus is considered to be as a genus name and Subtilis is considered to be as a species name. And we follow the laws of the binomial system of nomenclature, in which the genus name is always written as italicized or underlined. Uh, that is, the scientific name is written either italicized or underlined, and the genus name is considered to be capitalized, and the species name is never been abbreviated. That is, you can abbreviate means shorten the name. So always the genus name is written caps, and it can also be abbreviated. Now Bacillus is abbreviated as capital letter B here. The species name subtilis will always be written in small letters, and it has never been abbreviated. And sometimes you might also use the genus name alone to indicate a genus group, or a species name is never been used alone. So based upon these characteristics, we classify the organisms, and as well as we also name them. So I guess you got an idea of what is a microbial taxonomy. We would be going into the details of what are the characteristics with which we classify the organisms and the further details in the next session. Thank you for now.